This is the next video lecture on sexual reproduction in flowering plants. And in the last video lecture, we completed one of the important concept, that is outbreeding devices. What are outbreeding devices, and what are the outbreeding devices, and along with that, uh, outbreeding devices given in your NCRT. Those we completed in the last video lecture. And now in today's session we will study about the one of the important concept, that is pollen pistil interaction. Okay, before that I wanted to just recall your memory about outbreeding devices. And here, in case of outbreeding devices, mainly four they given in your NCRT. One is receptivity and synchronization, please remember. Second one is position of the flower or position of the reproductive organs. Then self incompatibility, this is also very important. And the next one, unisexual flowers, that means the production of unisexual flowers. These are the four important outbreeding devices. For what purpose these are? The outbreeding devices are developed in plants just for to avoid the self pollination. For what purpose? Just to avoid the that self pollination, and to avoid the inbreeding depressions. Okay, these are the four important outbreeding devices. This all points and everything we completed in the last class. And now we'll move on to the next concept, that is pollen pistil interaction. Pollen pistil interaction is also one of the important concept, please remember. And here, in case of pollen pistil interaction, what is going to be happen? And in pollen pistil interaction, usually series of events are going to be takes place here in pollen pistil interaction. And for what purpose this pollen pistil interaction? Here, in case of pollination, what will happen? The pollen grains from the anther, they are going to be transferred to the stigma. Okay, that is the condition. And stigma will accept that. Stigma should accept that particular pollen grain. Already in outbreeding devices we studied, they mainly avoid self pollination and they will mainly promote cross pollination. They mainly avoid self pollination, they mainly promote cross pollination just to change their genes. Okay, because variations they need, for that variation purpose they will undergo cross pollination. Okay, in that several important devices are there, those studied in case of outbreeding devices. And now here, in case of pollen pistil interaction, here what is the concept about the pollen pistil interaction? Here, whatever the pollen grains are producing, they're all not accepted by the pistil. It means either they are from the same flower or either they are from the another flower of the different species, because plants will not accept the pollen grains from the different species. They require the pollen grains from the same species. For that, there is a interaction just to identify either it belongs to the same species or different, or just to avoid the inbreeding depression. For example, self incompatibility. Okay, for that purpose this pollen pistil interaction occurs. And here, the n number of enormous number of pollen grains are going to be produced here. Okay, lot of pollen grains are going to be produced here, but only few of them get chance for the post pollination events. They get chance in the post pollination events. Post pollination event is nothing but fertilization. They will produce a lot of pollen grains. Okay, but this pollen grains, whatever the pollination is happening, it is not between the same species, either it is from the different species. Okay, it may occur between the different species, that will not going to be undergo the post pollination event if the pollen grains are from the different species. Okay, at that time what will happen, the pollen grain will not accepted by the stigma. Okay, and they are going to be recognize it, they are going to be recognized, either they are from the different species or they are the wrong pollen grains, or either just to avoid the self incompatibility. For that purpose there is an interaction between the pollen and pistil. There is an interaction between the pollen and pistil, and this interaction is mediated by some chemicals. Okay, because of some chemicals there is a interaction can occur. Okay, there is an interaction can occur just to recognize either it is a different pollen grain, or to just to avoid the self, just to take the self incompatibility. For that purpose there is a lot of events are going to be happen between the pollen grain and the pistil. Okay, but in case of male, in case of animals, what will happen, the direct interaction can occur between the female gamete and the male gamete. The sperm can recognize the egg it belongs to, is either it is the same or different. But in case of plants what will happen, it is not possible. Stigma is going to be interact with the pollen grain. Okay, stigma is going to be interact with the pollen grain and it will finalize that particular pollen grain. Okay, and if you observe the structure of pollen grain, if you observe the structure of pollen grain, this is the pollen grain. Okay, if you observe the structure, you can see here, this is the structure of pollen grain. Okay, this is structure of pollen grain, and here you can find the germ pore. Here there is the presence of germ pore, and here two types of cells we can see here. One is vegetative cell, another one is generative cell. Generative cell. And here from the generative cell there is a germ pore, and then through the germ pore there is development, that we'll study later. And when they recognize the particular pollen, they will allow, if the pollen grains are from the same plant or from the same species, they will allow the pollen grains to develop, and then they allow the germination, and then they will allow for the pollen tube growth, and finally there is a fertilization, and that pollen tube will reaches to the ovary, and then there is a fertilization. Like that there is a process is going to be happen. But what will happen if the pollen grain is from another species, or some plants will undergoing the self incompatibility? Okay, incompatibility. In that cases they are going to be terminate, or they are going to be inhibit, they are going to be inhibit the pollen germination, or they will inhibit the pollen tube growth on the stigma. Okay, they will inhibit the pollen tube growth on the stigma. How it is all thing happen? Just because of the some chemical interactions. Okay, pollen grains and stigma, they are going to be secrete some chemicals. With the help of those chemicals, with the help of those chemicals, there is a, with the help of those chemicals there is a interaction occurs. Okay, continuous dialogue you can see here, and because of that there is a selection of the particular pollen grain. Okay, there is a particular pollen grain. How they are going to be inhibit? With the help of some chemical reactions. With the help of some chemical reactions they are going to be identified, and if they belongs to the same species, there is a fertilization, there is a continuation of the pollination or fertilization occurs, and if they are from the different species, they are going to be reject. Okay, this is the thing, that how the pollens are going to be recognized, how the pollens are going to be recognized by the stigma. And if you take the diagram of that particular stigma, if you observe the if you observe the diagram of particular stigma. Okay, this is the complete structure of pistil. This is the complete structure of pistil. Here it is the stigma. Okay, this is the style and everything. Just consider it as a complete female reproductive part. And when the pollen grains come in contact with this stigma, when they will come in contact with stigma, it means when the pollination occurs, at that time what will happen, the right pollen grains will undergo germination. The right pollen grains will undergo germination, and wrong pollen grains are going to be terminate. And when they will undergo the germination, pollen tube is going to be develop here. Pollen tube is going to be develop here. How? Like this. Okay, there is a development, and the ovary usually present here. Here you can find the ovary, and inside the ovary you can find the embryo sac. And when there is a pollination occurs, and then if they are the right pollen grains, then germination occurs, and along with that germination there is development of pollen tube, and this pollen tube will run and reaches to the ovary, and finally fertilization occurs. In case of here stigma, they run along here, they run along the style here. This is the style you can see here, along the style, and then finally they reaches to the ovary, and inside the ovary there is a presence of ovule, and here there is the embryo sac, and there is a fertilization occurs, and further development is going to be happen. Okay, in case of right pollen grain, if the pollen grain is right, these all things are going to be happen. Okay, and when you come to the next aspect, that is, for example, just recall your memory. During pollen grain we studied that in microsporogenesis. Okay, in that what happens, if the pollen grains, just wait, if the pollen grains are first, they are going to be released from the anther. Okay, think it is a bilobed anther. This is the bilobed anther. The pollen grains are going to be shed when the dehiscence occurs. At that time the pollen grains are going to be shed out. They will come out from the anther. Okay, they will come out from the anther. Either they are two celled stage, in some cases two cell stage, either they are three celled stage. Either they are two cell stage or either they are three cell stage. Majority they are two cell stage. And what is two cell stage and what is three cell stage? Listen here, already we studied about the structure of pollen grain. There is a presence of two cells, one is vegetative, another one is generative. Vegetative cell will give the nourishment to the developing pollen grain. Okay, and here what happens, some of them are going to be shed into two cell stage. Two cell stage means there is only vegetative cell and then generative cell. But in case of three cell stage, okay, what will happen, generative cell will undergo mitotic division. The generative cell will undergo mitotic division. Okay, one generative cell, it becomes two. There is a formation of two male gametes. There is a formation of two male gametes. Okay, two male gametes are going to be produced over here. Two male gametes are going to be produced over here. In some plants what will happen, these two celled pollen grains are going to be shed from the anther, and then they will settle on the stigma. When they come in contact with the stigma, at that time they will undergo mitotic division, and the two cell stage become three cell. It means the generative cell will undergo mitotic division, and there is a formation of two cells, not three, two cells. Okay, two cells are going to form. But in some cases what happens, if they are three cell stage, when they are shed from the anther as a three celled stage, at that time what will happen, the generative cell already it completed its mitotic division. Okay, two gametes are already formed over there, and those two gametes or complete 3 cell stage is going to be shed from the anther, and then they will reach to the stigma, and there is no further development or there is no further mitotic division occurs over there, because it is already undergone mitotic division. It means there is already two cells are there, already two male gametes are there, so no need to worry about that. And when the germination starts along with the pollen tube, these all are going to be reach the ovary. When the pollen tube undergo germination, and then there is a development of pollen tube, and that pollen tube reaches to the ovary, and along with the pollen tube there is a transfer of the gametes. Okay, there is a transfer of the gametes. Okay, gametes are going to be transferred along with that. Okay, this is the what pollen pistil interaction. And one more thing we have to discuss here, that is uh, first we will settle on the stigma, and then pollen germination occurs, and finally there is a formation of pollen tube, and that pollen tube will run along the tissues of the stigma. Along the tissues of the stigma they are going to be run, and after that they finally reaches to the ovary, and then fertilization occurs here. Okay, fertilization occurs here. But when you observe the structure of megaspore, okay, when you observe the structure of megaspore, sorry, just wait. When you observe the structure of megaspore, you can see here different types of cells we can observe here. There is a microphyll, sorry, there is the ovals, sorry. When you observe the structure of this female gametophyte, there is a total 7 cells are there, sorry, no, 8 cells are there, and among these some of them are present in the chalazal end, some of them are present in the microphylar end. These are antipodal cells, and these two synergid cells, and here these two are polar nuclei. Okay, when the pollen grains transfer, when the pollen grains settle on the stigma, and then they will start to germinate, and they run along the tissues of the stigma, and finally they will reach to the ovary. Finally they are going to be reached to the ovary. And before that, first they will reaches to the microphylar end. This is the microphylar end. First they will come and reaches to the microphylar end, and there is no chance of entering into the chalazal end or any other side. Okay, they will directly comes, or they directly enters to the microphylar end. And when they enters to the microphylar end, there is a presence of filiform apparatus. There is a presence of filiform apparatus here. Okay, and these filiform apparatus will guide the pollen tube. Okay, they will guide the pollen tube to reach the synergid cells. These are the synergid cells, just to reach the synergid cells. And next to the synergid cells there is a presence of egg cell. Okay, so there is a guidance that is mainly given by the piliform apparatus. And then those gametes, whatever the gametes are present, they traveled along the pollen tube, they enter here, and then they are going to be fertilized with egg cell, and remaining double fertilization and everything that we will study in later classes. Okay, we'll study in the later classes. Okay, this is the thing, how the things are going to be happen, and then completely in detail we'll study in the double fertilization. Okay, just remember this much only they given, just remember this one. Okay students, I think so you understood the concept. What is the what I have explained still now. Stigma, when the pollen grains are come and settle on the stigma, they will choose the correct pollen. If they are wrong pollen, they will inhibit or they will stop the pollen germination and pollen tube growth. But if they are right, they will allow for pollen germination and they will allow for the development of pollen tube, and that will reaches finally to the ovary. Then fertilization occurs. This how it is going to be mediated. This is going to be mediated by some series of chemical reactions. Okay, chemical reactions. Chemicals are going to be do this mechanism. Okay, and how they are going to be transfer here. Pollen tube will run along along the stigma, tissues of the stigma, and then finally reaches to the ovary. And in the female, there is in the female gametophyte there is a microfilament, and in that microfilament there is a presence of piliform apparatus, and those will piliform apparatus will guide the pollen tube or the gametes to reach the synergy, and the synergy will guide to fertilization. Okay, this will happen in case of pollen pistil interaction. And this germination of the pollen tube, germination of the pollen tube and so germination of the pollen grains and then development of the pollen tube will study in the practical. Okay, with practically we'll going to be study about that. Okay, what we are going to do in the practical, just we'll add 10% sucrose solution to the pollen grains. Just we'll collect the pollen grains from the different plants. Say for example pea plant, we can take the pea plant, we can take the chickpea plants. Okay, we can take chickpea plants, we can take vinca rosea, we can take hibiscus. Okay, we can take hibiscus. Okay, commonly we find the hibiscus and the vinca rosea. Okay, we'll go through that vinca rosea and hibiscus, and vinca rosea is the easier one that we will see in the practical. Okay, in that what we are going to do, just we will collect the pollen grains and we will add 10% sucrose solution to the pollen grains, and we will wait for uh, 10 to 15 to 20 minutes. We will wait for 15 to 20 minutes, and after that if you observe under the microscope, we will find the pollen germination very clearly. We will find the pollen germination very clearly. Okay, and that we will study in the practical. Okay students, this is all about the pollen pistil interaction what we studied till now, and some points I was given here. These are all the points what I was explained till now, and for further information or you can refer other books or you can refer NCERT. Go for NCERT, and that is also very nice one. You can read NCERT repeated times, and you can go for other books. Okay, for NEET books and other books, and then that also you will find a huge amount of information. And this is what I was explained. This is in your NCERT textbook. Okay, this is in your NCERT textbook. You can refer and you can follow. Okay, these are all the points, very simple points are there. Please remember, for competitive examination point of view it is very important, and sometimes in board examination also they will ask the question. Okay, very simple, please remember this one. And in the next video we'll discuss about artificial hybridization and double fertilization. Okay, in the next video lecture we'll go for artificial hybridization along with double fertilization. Okay, and this is about the pollen pistil interaction, students. And if you have any queries or any doubts, you can ask me through WhatsApp, or you can add comments comment to this video. Okay, thank you, and we'll meet the next video lecture. Thank you.